Large-scale production of yeast. Yeast constitute an interesting group from a technical and industrial standpoint in microbial world, and it has several uses in different types of industries. Yeast are generally found in soil, in curd, various foodstuffs, fermented foodstuffs, and it can be very easily isolated from aerial surfaces of plants, mainly from grapes, vegetables, pomegranates, etc., using suitable media. There are several commercial applications of yeast, as we have learned earlier, that is mainly in the production of alcohol and alcoholic beverages, baking process, foodstuffs, animal feed, and production of different biochemicals. Among these, Saccharomyces cerevisiae is considered as the most versatile organism. It is used in several of these processes. Depending on the commercial application of yeast, there are several categories. Yeast which are used in the baking process are called as baker's yeast. Then in ethanol production will be called as distiller's yeast. Production of food yeast, which has application as uh, a food supplement for human consumption. Food supplement for animal consumption, it will be called as fodder's yeast. The method of production. For the production, large scale production of yeast, general composition of the medium is that it should contain fermentable carbohydrates, a source of nitrogen, trace elements, phosphates, sulfates, and vitamins. Along with that, it should contain several growth stimulators that includes amino acids, inositol, thiamine, pantothenate, riboflavin, and different types of uh, vitamins. Several media may constitute of brewer's wort, grape must, and apple juice that serves as a source of these different materials. For the commercial production, the medium can be prepared by using several basal media, for example, molasses based medium, cheese whey based medium, sulfite liquor based medium, and petroleum based medium. The use of medium is dependent on the variety of yeast which is to be propagated. For example, molasses based medium is basically useful for the large scale production of baker's yeast, food yeast, and distiller's yeast, whereas cheese whey medium is useful for the large scale production of Cluromyces fragilis and Candida pseudotropicalis, which are the food and fodder yeast. Basically, these yeasts are a lactose fermenting yeast, so it is more. This will be the most suitable media as it is rich in lactose. Whereas for the large scale production of Candida utilis, it may be sulfite based liquor that has an ability, the Candida utilis has an ability of utilization of a wider range of sugars. Candida lipolytica, it is mainly known for the assimilation of hydrocarbons like N-alkanes. Therefore, the most suitable medium for its large scale production will be using petroleum fractions. A production of yeast is a multi-stage aerobic fed batch process. In general, this production plant may consist of fermenters which are connected in series. In this figure, I have just shown only four fermenters which are independently operated. Uh, all these fermenters are sterile closed fermenters which are connected in series. As the name indicates, that it is a fed batch process. We have learned about batch fermentation, we have learned about continuous fermentation, and the fed batch, it comes after the batch fermentation. The simplest meaning of fed batch is uh, we have to feed a batch fermenter. That's why it is called as fed batch. That means there is intermittent addition of fresh nutrient medium or substrate. Intermittent means after a periodic interval there is addition of fresh medium. So in the first fermenter, that is F1, sterile nutrient medium, one third of sterile nutrient medium is added, and then it is inoculated with the stock culture of yeast. which is then repeated, which is the same process is repeated for F2, F3, and F4. Since it is aerobic process, there should be vigorous supply of oxygen, which is one of the essential factors. Here, the major objective is the propagation of yeast. Yeast are aerobic organisms. Uh, if the supply of oxygen is less in this case, then the metabolism may get diverted towards a fermentation, and instead of getting a huge biomass, there may be a production of alcohol or different type alcohol like substances. So in all these fermenters, there is periodic addition of nutrients or substrate unless the final volume is achieved. And at the end of the fermentation, the aeration is stopped, the addition is stopped, the aeration is continued for next half an hour or 1 hour or so, and then the fermentation broth is separated from all these fermenters at a time for the recovery of the microbial biomass, yeast biomass. Production of brewer's yeast. For production of brewer's yeast, steam sterilized production plant is used, uh, in which there may be addition of pasteurized hop malt wort or the boiled malt. Remember that this boiled malt is basically it contains all the essential nutrients which are required for the brewer's yeast. A typical system consists of two propagating closed vessels, fermentation vessels, and each vessel would be inoculated with 100 grams of precis, that is the inoculum from a laboratory stock. Then there is continuous aeration at temperature of 20 degrees Celsius. This process is going to generate a lot of heat, as there are going to be exothermic reactions, and therefore this closed fermentation vessel has to be equipped with exterior cooling coils through which the cooling water it flows, and it helps in the control of temperature. Then the yeast mass it exponentially it increases to give approximate 17 gram press yeast per liter after 48 hours. This culture will be then useful for this will be the production of brewer's yeast, and this brewer's yeast which is propagated as shown above, is it will be useful for the inoculation of fermentation vessels, that is for the brewing process. Production of a baker's yeast and distiller's yeast. Baker's yeast is propagated on beet molasses or cane molasses or blackstrap molasses based medium. The molasses is diluted with water, pasteurized, blended, and supplemented with essential compounds such as the source of nitrogen ammonia and ammonium sulfate, diammonium hydrogen phosphate, and magnesium sulfate. A small amount of production medium is added into a sterile closed production fermenter. Inoculation, which is then followed by inoculation with cream yeast or stock yeast, which is propagated fermentatively in successive seed fermenters from laboratory culture. Then there is aeration, that is supply of sterile air. There is intermittent feeding of fresh medium into vessel, that as we have learned earlier that it is a fed batch process. So it is called as a fed batch process. So it is necessary fed batch, that means intermittent addition, periodic addition of nutrients is essential, since as the high concentration of sugars like sucrose, glucose, or fructose, so these sugars which are present in molasses are responsible for repressing the respiratory enzymes. So high concentration it represses the respiratory enzymes, and if the respiration process is hampered, the biomass, the building up of biomass of yeast, it will also get suppressed. So in order to get higher amount of biomass, these sugars should be added in this sugar should be added intermittently. That means periodically, so that the final concentration should not exceed, and there should not be repression of respiratory enzymes. That is one of the reason why the fed batch process is important process of production of yeast. To keep the ratio of cell mass, this is also essential to keep the ratio of cell mass and sugar constant. Accordingly, the rate of aeration is adjusted. During this process, there is going to be a lot of foaming. Therefore, there is use of antifoam agents like polypropylene glycol, silicones. There is going to be production of heat which can be controlled by the circulating coils. The pH is kept in the range of 4 to 4.5, which can be maintained by the addition of several neutralizing agents. Finally, after the incubation period is over, the addition of nutrients is stopped. The period, that means the fed batch, a feeding of nutrients is stopped, and the aeration is continued for next 30 minutes. This after feeding is stopped, the further incubation is essential because that will allow the cell division to complete. Complete cell division means formation of complete yeast cells, no presence of budding cells. So this continuous aeration, it will help to reduce the number of budding cells. The final culture mainly contains approximately 50 grams of yeast dry matter per liter. The recovery, it involves separation and clarification of yeast. So yeast are separated, the liquid bulk liquid is separated. It will be thick in nature. It will be called as yeast cream. So this yeast cream is washed for the separation, for the clarification of yeast cells. That means for the uh, separation of any coloring agents which are contributed by the media such as molasses. So yeast cream is frequently washed with water, centrifuge. Again, it is mixed with water, washed with water and centrifuge. So there are several cycles of washing with water and centrifugation that will helpful for the clarification or the separation of coloring matter. Then the next step is dewatering. That means this is a method of concentration that includes drying of the product on equipment called as rotary vacuum filters, as we have learned in the last semester. A drier product containing 30% of dry matter content, it can be obtained at a temperature of drying at temperature of 45 degrees Celsius. Then the blocks of the yeast are prepared, and these blocks are stored can be stored at low temperature. From this yeast preparation, it can be further pressed for the separation of water that will give a consistent product having percentage solids of 29 to 31%. It will be called as compresses. Or this yeast mass, it can be extruded through a screen in order to produce a homogeneous product, and it is chopped. Finally, it is dried. The pellets are dried in rotary dryers for 6 to 24 hours at 45 degrees Celsius, so as to obtain the product called as active dry yeast. Similarly, another formulation, it can be a yeast formulation, it can be prepared. So this formulation, it can be diluted with water in order to form yeast cream, so that the final preparation contains 17 to 19% of solid. So these are the several formulations of yeast which can be prepared and which can be stored at low temperature unless it's used. Remember that Saccharomyces species, that is distiller's yeast, produced on molasses media for the use in ethanol production, that is in distillery, are produced in the same way as the pressed baker's yeast.